We're gonna begin where fertilization and implantation occurs. Here is female reproductive tract. This is vagina, cervix, uterus, ovary, fimbriae, which is a part of fallopian tube, and this is fallopian tube. Basically, female release egg per every month during their menstrual cycle, which is known as ovulation. Here is the ovulation occur, which is collected by the fimbriae. Oocyte, which is surrounded by thick protective layer, which is known as zona pellucida. And surrounding the zona pellucida there are structures, cells, which is known as coronary radiata. This is the basic structure of the oocyte. If this oocyte is not fertilized by a sperm, periods occur. But if there are a lots of sperm swimming in uterine tube and then oocyte and sperm mates, this process is known as fertilization. Here is also zona pellucida is present. Structure. Coronary radiata is also present here. Here is the sperms. The sperm is able to penetrate zona pellucida with a special enzyme which is present on his head. Anyway, once fertilization occurs, a zygote is formed. Here also zona pellucida is present. This is known as zygote. We can also say fertilized egg. The sperm DNA and oocyte DNA mixed to form literally one cell. Then that one cell then divided into two cells. Then two cells undergo successive mitotic division to form 4, 8 and 16 cell division. Here is 2 cell, here is 4 cell, and here is 8 cell structure. This process is known as cleavage. Then again 8 cell structure undergo mitotic division to form 16 cell structure. Here is also zona pellucida is present. This structure is known as MORULA, 16 cell. Then again MORULA undergo to form 32 cell structure, which is known as BLASTOCYST, 32 cell. Let's talk briefly. First of all, cleavage. What is cleavage? Cleavage is the process in which one cell structure undergo successive mitotic division to form the 8 cell structure. Here I am drawing one cell structure undergo mitotic division. First mitotic division to form the 2 cell structure. Then again 2 cell structure undergo mitotic division to form the 4 cell structure. Again, 4 cell structure undergo mitotic division to form the 8 cell structure. 8 cell structure. So basically, what is cleavage? Cleavage is the process in which one cell structure undergoes successive mitotic division to form the 8 cell structure. Again, 8 cell structure undergo further mitotic division to form the 16 cell structure. It undergo mitotic division again. Mitotic division to form the 16 cell structure. The 16 cell structure is known as MORULA. MORULA consists of two types of cells. Outer and inner. The inner cell is known as inner mass cell and outer is known as outer mass cell. Zona pellucida is present here. Then again, morula undergo mitotic division to form the 32 cell structure, which is known as blastocyst. This is zona pellucida. That is the outer membrane of the blastocyst. This outer membrane is known as trophoblast. And here, as you can see, inner mass cells gathered to form grape-like structure. These cells are known as blastomeres or embryoblast. There are two poles. Point at which embryoblast gathered is known as embryonic pole, and opposite it is embryonic pole. Embryonic pole attached to the endometrium of the uterus for the implantation. Now you can see here is a cavity. This cavity is known as blastocele. Then what is the function of the trophoblast and blastocele? Trophoblast helps in the formation of placenta and provides nutrition. And what is the function of the blastocele? Blastocele, it permits the cell, it basically permits cell migration during the gastrulation. This is all about the first week of embryogenesis. I hope you liked the video. If you like the video, like, comment and share the video. Thank you so much.